feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, hello, and welcome to the National University of Singapore Yongloo Lin School of Medicine's Healthy Longevity webinar. Thanks for joining us tonight. Dr. Kristin Gribble will be with us tonight. She has a very interesting talk for us, but before we get to that, I would like to do two things. One, to remind you to use the Q&A function to send questions. And second of all, I'd like to introduce Matthias Robert, a research assistant in the U.S. Center for Healthy Longevity, who'll be talking about his study, therapy-induced senescent tumor cell-derived extracellular vesicles differentially suppressed with progression.
Thanks for the introduction. As mentioned, my name is Matt, and today I will be talking about therapy-induced senescent-derived extracellular vesicles. It is widely known that cancer treatment itself could cause cellular senescence, which is termed as therapy-induced senescence, or TIS. When cells become senescent, they secrete a bioactive component called the senescence.
I apologize. That seems to have gone wrong. I can try and get him back on again, but I'm going to put you back to Brian. Brian, you okay there?
Yeah, I'm here. And I'll wait just a few minutes to see if we can get the rest of that back on. If not, we can go on to the talk. So
I don't think I can recover this right now. I'm so sorry about that.
I want to introduce Kristin Gribble. She received her PhD from the MIT Woods Hole Oceanographic Institution Joint Program in Biological Oceanography. She's currently an associate scientist at the Marine Biological Laboratory, and her work is on maternal age effects, and that's funded by both the NIH and the NSF. She's also the recipient of the NSF Career Award and a fellow of the American Aging Association. The topic of her talk tonight will be the effects of maternal age on offspring health and lifespan. Thanks for joining us, Kristin.
Hi, thanks so much for having me here today, and thanks for that introduction. I'm going to go ahead and share my screen here and get started. So today I'll be talking about the effects of maternal age on offspring health and lifespan, which is what we work on in my lab. My lab is at the Marine Biological Laboratory, which is in Woods Hole, Massachusetts, on the east coast of the US, just south of Boston. We're a small private research institution studying all aspects of biology. So I wanted to start by just defining what I mean by maternal effects, because I'll be talking about maternal age effects. So maternal effects are when an offspring's phenotype is determined by its mother's environment or physiology. And from the biomedical literature, we know of lots of examples of negative maternal effects. So, for example, excessive smoking or drinking during pregnancy can lead to negative cognitive or metabolic outcomes for children from those mothers. Another example is that of the Dutch hunger winter, where during World War II, a German blockade of the Netherlands caused widespread famine over a winter. Women who were pregnant during that time or who became pregnant shortly after that famine their children as adults had much higher levels of neurocognitive deficits and metabolic diseases. So we know of lots of negative maternal effects from the biomedical literature. From the ecological literature, however, we know of many examples of beneficial maternal effects. So, for example, in this zooplankton species, Daphnia, small little invertebrate, when mothers are exposed to predators, their offspring hatch with these large defensive spines, head spines, and tail spikes that are effective in defending against those predators. So in my lab, we're interested in both potential negative or beneficial maternal effects, and we're specifically studying maternal age effects. So this is when the age of a mother at the time that her offspring are born affects the phenotype, the health and lifespan of her offspring. People have known about maternal age effects for a very long time, actually. For example, in this monograph published by Alexander Graham Bell in 1918, where he studied the genealogy of the Hyde family, this very extensive genealogy encompassing thousands of people, Bell found that offspring from mothers over age 40 had on average 10-year shorter lifespan than children from mothers who were 25 or younger. Probably if you see in this figure, there are very short lifespans. Probably a lot of that was caused by infant mortality at that time. But still, these findings based on thousands of individuals are really pretty striking. One of the first examples of studying maternal effects in the laboratory was done by Albert Lansing in the 1940s. He used another small invertebrate, rotifers, and created lines of constant maternal age. So successive generations were derived either from all young mothers for many generations or from all old mothers over many generations. And he found that lineages that came from successive generations of old mothers had much, much shorter lifespan and lower reproduction than those that came from young mothers over many generations. And so these negative effects of advanced maternal age are often referred to as Lansing effects. Since that time, there have been many other examples across a very wide range of species in which we see that offspring from older parents have much shorter lifespan than offspring from young parents. And this ranges from nematodes, C. elegans, Daphnia, fruit flies, up into mammals and including humans. So this is a very widespread phenomenon across the tree of life. Like I said, there's evidence of negative effects of advanced maternal age on offspring in people, but it's really inconvenient to study this phenomenon in humans, right? We live for a very long time. We're genetically diverse. We're not allowed to, nor should we experiment on pregnant mothers or their children. And so we move to other model systems, animal systems, to study maternal age effects. In my laboratory, we're using rotifers as our model system. Rotifers are microscopic aquatic invertebrate animals. So they're little zooplankton. There are actually 2,000 different species of rotifers that have been defined. You can see in these examples, they vary greatly in their shape and size. But you can find rotifers pretty much anywhere that you can find water. This includes bays and lakes and ponds, rivers, but also includes bird baths, puddles, wet moss on trees, literally anywhere there's water, from the poles to the Arctic. And rotifers are actually quite important ecologically, which I won't talk about today. So in my laboratory, we're using this species of rotifer, Brachionis mahavacus. This is a female Brachionis. You can see here at the top end of this animal, which is about 0.5 millimeter in length. There are cilia that are used for swimming and filter feeding. There's a large jaw in this area that's used for grinding food. The gut of this animal is packed full of algae, which is what the rotifers eat. And this female is carrying these 4 very large eggs, and we'll be talking more about those large eggs. Off to the side here is just an appendage that's referred to as a foot with these 2 little toes on the end. So rotifers swim around. They can also attach to surfaces. They have pretty complex behavior. So why would we use rotifers to study something like maternal age effects? They actually have a number of advantages, like other established invertebrate model systems that are used to study aging. Rotifers are simple to culture. They have a short lifespan of just about 2 weeks, so we can very rapidly do aging assays. We can work with individuals, up to millions of individuals per experiment. As you can see in these pictures, they're transparent, which makes it easy to image morphology and cellular processes. And now we have really growing genomic and transcriptomic tools available for rotifers that include sequenced genomes and transcriptomes, RNAi, and CRISPR to do genetic manipulation in these animals. Rotifers have some advantages over some other model systems, which makes them, we think, fairly unique. We know a lot about the ecology and life history of rotifers. People have been studying rotifers for over a century. So we really know a lot about how they behave normally. Rotifers have direct development. That means there's no larval stage and no metamorphosis. When a rotifer hatches, it's just a small rotifer that hatches out of the egg, turns into a larger animal. So a little bit more similar to what happens with people, right? There's no metamorphosis. And so that means when we're doing our lifespan studies, we're studying the entire lifespan of the animal, not just the adult lifespan. These rotifers don't exhibit any sort of parental care after their offspring hatch, which allows us to separate questions of nature versus nurture as we're studying maternal age effects. And these animals make a very large investment in each individual offspring, again, somewhat similar to what humans and other mammals do. So instead of having hundreds or thousands of offspring over their lifespan, like many other invertebrates do, these Brachionis mahavacus rotifers produce about 25 individual offspring that are quite large when they hatch over their lifespan. And then very importantly, rotifers share more genes in common with humans than do some other invertebrate systems. Worms and fruit flies have lost a lot of genes over evolutionary time that humans still have. Rotifers seem to have held on to those genes. So they have literally hundreds more aging-related genes that they share in common with people than do some other laboratory invertebrate systems. So we think together these make them really a useful laboratory tool to understand the basic biology of aging. So rotifers do have a really nice aging phenotype. They hatch as small little individuals. We call them neonates. They start reproducing at about 1 to 2 days old. There's a peak in reproduction quite early in life. You can see that here, this purple line with increasing age. And then reproduction declines as the rotifers age. And these rotifers have a short post-reproductive period of a few days before death at an average of about 2 weeks old. We also see that measures of health change with age in rotifers as they do in other animals. So we can't ask a rotifer how it's feeling or as it's getting older. So we use a number of different assays to try to assess health. One of these is phototaxis. That's the tendency to swim toward a light source. So phototaxis declines with increasing age in rotifers. Resistance to heat stress declines with increasing age. Swimming speed declines actually quite early in life and then remains low into late age in rotifers. And behavior additionally changes with increasing in late age, so that older rotifers are more likely to be sitting still rather than swimming, where younger rotifers spend much more time actually swimming around. So we use these sorts of assays in our studies of maternal age effects in rotifers. We're just looking here at a female rotifer, again with this one with 4 eggs and relatively newly hatched neonate rotifer that hasn't begun reproducing yet. So to do these maternal age effects in these animals, we begin with a cohort of age-synchronized females. We follow these individuals over their lifespan and collect offspring from young, middle-aged, and old mothers, and then measure whatever it is we're looking at in those offspring from mothers of different ages, lifespan, or measures of health typically. And what we see is that offspring from older mothers in this species of rotifer do have shorter lifespan and lower reproduction than do offspring from young mothers. We see that here. These are offspring from 3-day-old mothers, 7-day-old mothers, or 11-day-old mothers. So young mother offspring are in black, old mother offspring are in blue. And we see about 25% lower median lifespan in old mother offspring compared with young mother offspring. Total lifetime reproduction is also lower in old mother offspring. In this particular experiment, about 10% lower in old mother offspring. Maternal age also affects offspring health. So we see here this phototaxis measure, swimming toward a light source. Young mother offspring maintain a high level of phototaxis throughout life, where in old mother offspring, phototaxis declines much more rapidly as those individuals age. Not all measures of health are worse in old mother offspring, however. We were very surprised to see that heat stress resistance is actually improved in old mother offspring compared with young mother offspring throughout life, from early life to late life. Right, so we see that lifespan, reproduction, and certain measures of health are lower in old mother offspring compared with young mother offspring. We also see that maternal age effects and environmental effects can interact in these animals. And the example of that I'll show today is that of caloric restriction. So if you've been to these webinars before, probably you've heard about caloric restriction studies at some point in the past. But we do see in most species that have been studied before that lower levels of food lead to increased lifespan across a wide range of species. And that's true in rotifers as well. We've done a number of studies of caloric restriction in rotifers. We do that by feeding some rotifers full food under full food conditions, which we call ad libitum conditions, under chronic caloric restriction, which is just constant low levels of food, or under intermittent fasting, which in our case is every other day, high food and no food feeding. Of course, we don't feed the rotifers hamburgers. We feed them single-cell phytoplankton under those different food conditions, ad lib, chronic caloric restriction, or intermittent fasting. And we have seen in multiple experiments that both chronic caloric restriction and intermittent fasting increase lifespan in Brachionis mahavacus. But when we combine maternal age effects and caloric restriction, we see something fairly interesting. So in some experiments, we have imposed caloric restriction on the mothers and then collected offspring of from mothers of different ages and fed those offspring all under full food conditions. And what we see is that the offspring from older mothers have a greater benefit from maternal caloric restriction. So these offspring are not calorically restricted themselves, but offspring from old mothers, if those mothers had been either subjected to chronic caloric restriction or intermittent fasting, old mother offspring have increased lifespan, increased reproduction. Interestingly, they also have increased body size. This is something we see across in offspring from older mothers, is that they typically have much larger body size than offspring from young mothers. And then if we do the reverse, where we feed mothers under full food conditions and then subject offspring to either chronic caloric restriction or intermittent fasting, we see that old mother offspring derive greater benefit from caloric restriction than do young mother offspring. So here, young mother offspring under intermittent fasting have about a 17% increase in median lifespan, where old mother offspring under intermittent fasting have about a 50% increase in median lifespan under that intermittent fasting. Right, so environmental effects and maternal age effects really do interact to determine offspring phenotype. And what we've seen in these caloric restriction experiments is that old mother offspring have the greatest relative benefit from caloric restriction, greater lifespan increase, and a greater increase in lifetime reproductive output. Right, so what I've shown you so far are phenotypes of maternal age effects, whether it's decreased lifespan or decreased reproduction in old mother offspring, or changes in responses to age-related therapies, or changes in measures of health. It's all really interesting, but of course what we would really like to know are what the mechanisms are that drive, that cause these maternal age effects. We think that if we can understand these, it will help us understand some basic biology of aging, and also help us to understand how we might mitigate some of these negative effects of advanced maternal age. Right. There are probably lots of mechanisms at play here. One thing that we think about frequently is how maternal investment in developing offspring may change with maternal age. We've measured this in a few different ways. We've looked at egg size, lipid content in eggs, and then starvation resistance in offspring from different age mothers. We do see changes in maternal investment with increasing maternal age. So egg size, interestingly, increases with increasing maternal age, but lipid content decreases with increasing maternal age. And we also see that starvation resistance is lower in offspring from older mothers, suggesting that those offspring from older mothers are coming into life with fewer energy stores than offspring from young mothers do. There certainly are effects here, but they're significant, but they are relatively small. It's hard to know how biologically meaningful they are, and they don't seem to be big enough to account for the very large changes that we see, differences that we see in survival and fecundity in old mother offspring. So one of the things that we're investigating is the potential role of mitochondria in maternal age effects. There are many good reasons to think that mitochondria may be involved in these non-Mendelian effects that we're seeing of advanced maternal age. Mitochondria are, of course, the powerhouse of the cell, right? They control largely energy production, but they're also involved in calcium homeostasis, redox homeostasis, stress response, and so on. Mitochondria are also maternally inherited. Mitochondria have a genome that's independent of the nuclear genome. That genome, mitochondrial genome, is present in many, many copies per mitochondria, with many mitochondria per cell. And those numbers, both genome copy number and mitochondria number, are dynamic over time. And so we think that that mitochondria could be very important in controlling some of these maternal age effects that we're seeing. And so we're investigating multiple aspects of mitochondrial phenotype in offspring from different age mothers. And we do see differences in mitochondrial shape and size with increasing maternal age. Very large differences in mitochondrial metabolism, which we're measuring using targeted mitochondrial metabolomics, and quite large changes in gene expression related to mitochondrial function. I'm not going to show a lot of data, or really any data, from these studies at the moment. This is all still relatively preliminary, but this is what the lab is actively working on right now. And we are seeing that it's quite likely that mitochondria are playing a large role in propagating these maternal age effects. Right, so since we're pushing up against time here, just to wrap up, I just wanted to remind you that what I've shown you is that offspring lifespan, fecundity, and health decline with increasing maternal age in these rotifers and in many other species as well. We do see that offspring response to either their environment or to interventions, or therapies, can vary with maternal age. Increasing maternal age is associated with changes in offspring mitochondrial function, morphology, and metabolism. We think that plays a really important role in causing these maternal age effects. And I think it's important to understand that maternal age may be a cause of a lot of uncharacterized variability among individuals with aging, right? We know that individuals really vary a lot in in how they age. Their health and lifespan is really different between individuals. And I think differences in maternal age could be one source of that variability. And with that, I just wanted to thank folks in my lab who did this work, both past and present lab members, collaborators, both in the US and other institutions, and then our funding sources. And with that, I'd be happy to take questions.
Thanks a lot. First of all, it's the most beautiful organism, at least by those pictures.
Yeah, we like them.
It's also striking that, these are very small creatures, but they have a lot of different cell types. They have a nervous system, and I guess it's possible to look at the role of different tissues in the aging process.
Yeah, exactly. And that's something that we're just beginning to explore. So rotifers do have, their nervous system is actually about 25% of their cell number. Rotifers have 1,000 cells. About 250 of those are in the brain and peripheral nervous system. They have a digestive system, reproductive system. They have muscles that, under transmission electron microscopy, look very much like human skeletal muscles. Right, so we really can dive in and start to look at tissue-specific effects in aging.
I wonder, it struck me that the speed with which they swim drops off so quickly. Is that a reflection of an early decline in muscle function, or is there some other behavioral explanation for that? It seems like it's out of scope with the other phenotypes, and also it's a little different from mice, where things like sarcopenia don't appear until very, very late. So
Right. That's a really good question. That's not something that we or anybody else has looked at, I guess. I think rotifers really are programmed to live fast and die young. They in the real world live in very ephemeral environments, and they are food for everybody in those environments. So they're not likely to survive very long. So I think they've really evolved to do most of their important work early in life, right? We see that reproduction happens, most of it, quite early in life. And so I'm not sure if, the swimming is a reflection of that as well.
Interesting. I guess you said this and I missed it. There's asexual reproduction and there's sexual reproduction. So is it like worms? There are hermaphrodites and males? Is that the...
No, not exactly. So these rotifers normally reproduce asexually, so they're all female, and they undergo just mitosis in the germline, and so the offspring that are produced are clones of their mothers. In response to environmental conditions, some mothers will produce daughters that are sexual. Those daughters will produce males, or those sexual females can be fertilized. So there are no hermaphrodites. There are just females and males, and the females may be asexual or sexual.
So if you look at the offspring from the sexual reproduction versus asexual reproduction, how does that affect lifespan?
Lifespan is similar in the sexual females and asexual females. The offspring from sexual reproduction, those offspring are themselves asexual. So they are just like asexual females. The males, interestingly, have very short lifespan. The males have no mouth or gut, so they never feed. They're very small. They swim very fast. They have one job to fertilize females, and that's, yeah.
Can you look at things that affect lifespan in the males, or is it too hard to get a lot of them to do it?
It's actually quite easy to work with the males. They're simple in the laboratory because we don't have to feed them, which is nice. We've done a bit of work with males as well. We do see similar effects of maternal age on male offspring that we do on female offspring. What I showed just now, those were all female offspring. But we haven't done a lot else with males in terms of looking at, stressors or therapies that could increase lifespan in males.
I'm wondering, in the case of CR, you talk about it extending lifespan. Are there other interventions that extend lifespan as well, that either genetic or other strategies?
Yeah, so many. So rapamycin works quite well in rotifers. There have been a few studies published on that, mostly from Terry Snell's lab. We've shown that in my lab as well. There are a number of different drug interventions that work quite well in rotifers. We are currently doing screening in my lab of FDA-approved drugs that have targets that gene targets that seem like they should be involved in aging, to see if those already approved drugs may have uses in aging. Rotifers are, like C. elegans, a really nice screening tool for drug studies. So,
yeah. And other interventions, do they, like, you may not know this yet, but I'm curious whether there's something specific to CR in terms of, it almost as if it normalizes the difference between offspring from young and old parents. And so is that going to be true of every intervention, or is it specific to CR? Is that telling us something about metabolism that is relevant?
It's interesting. We haven't, say, in the drug studies, applied drugs to mothers and then looked at the effects on offspring. Most of our drug studies have been in one generation. I would guess that for certain drugs, potentially those that have an effect on metabolism or maybe epigenetic inhibitors, that those may have multi-generational effects as well. I don't really know what to say if it's metabolism related or not specifically. But
it seems an interesting question because the CR finding is really interesting, and if it's really a normalizing effect, or if that's just what you would see with every intervention, I think is if it's the former, I think that really is a clue to something about the mechanism from old mothers. And
Right.
I think also the size effect is interesting because so if I understood correctly, the offspring from the old mothers are larger,
Correct.
right? And the CR doesn't rescue that. It rescues the other phenotypes, right?
Right.
It's
Right.
interesting. Um, but when I think about larger, flies that are larger live shorter. A lot of species that are larger live shorter. And, that's been attributed in various contexts either to IGF signaling or mTOR signaling. So, and you mentioned already that rapamycin extends lifespan. I'm wondering if the mTOR pathway is elevated in the offspring of the old mothers.
Yeah, that's a really good question. So we have just done a very large transcriptomic study to look at differences in gene expression between young mother offspring and old mother offspring. And we're just starting to dig into those data to really sort of pull out the important pathways. But I think you're right that might be a really important one to look at.
So the offspring are larger from the CR, but the lipid content is less, is what you're saying, right?
Right.
So I guess there's a lot of lipid storage. Um, I'm trying to figure out what's replacing the lipids. You've got extra size, but what's in it?
Right. Yeah. So we don't know if there really just the cells are larger and what it would be in those cells that's making them bigger. Yeah, it's really unclear to me what would be causing that size difference. Yeah, so we've only measured the measure of lipids that I showed was just an imaging measure of lipid droplets. So we haven't gone in and done real lipidomics to look at shifts in lipid content or lipid composition in offspring from different age mothers. There's a huge range of, work still yet to be done to really characterize just.
You're in this kind of great space where you can almost anything you can think of is an interesting experiment. I remember when we started working on yeast aging, there were some studies already done, but pretty much anything we came up with was like, yeah, I don't know. I wonder what'll happen. It's a very fun time to be working in a laboratory because, there's just a wealth of good ideas, and I guess it helps, to get the resources to follow them up too. But
Yeah.
but it
Yeah,
seems like a great system.
Yeah, that's really the limitation, right? It's just people and time to do more studies. And now we do have tools available for these sort of non-model systems that weren't available even 5 years ago. So 5 or 8 years ago, we didn't have many sequenced genomes or transcriptomes, and we certainly didn't have CRISPR. And those will all really allow us to answer questions that we haven't been able to answer in the past.
Yeah. I mean, being able to just make any mutation we wanted to in yeast is what allowed us to go quickly. So I think that it'll really accelerate programs in species like rotifers too. So,
and I think this rotifer system is really easily adoptable by almost any laboratory. Rotifers are incredibly easy to grow in the lab and maintain. They're used as the species of rotifer is used as food in aquaculture systems. So zebrafish laboratories also often have rotifers that they are feeding to their larval fish. They're a nice size. They don't take up much space. You don't have to deal with, mammal protocols, IOCOOK or things like that.
you're starting to convince.
Yeah, I think we're in a position now where lots of other laboratories could adopt the rotifer system, which would be great. I'd love to have more colleagues in this space.
Quick question. Maybe we can talk about the human data just a little bit. I mean, how robust is that, and how big of a lifespan difference is there in sort of the modern human studies on this?
Yeah, there haven't been many studies, and there are epidemiological studies. There are all sorts of other factors that are that could contribute and sort of muddy the data. So they tend to be pretty small lifespan differences in offspring, children from older mothers. Yeah, so it's a little hard to know because you can't control those studies particularly well.
Yeah, it's a fascinating question though, because the age at which mothers are having babies is going up and up and up. And so it's and we're looking at strategies in our ovarian center to preserve fertility so women have more options and freedom to choose when they want to have babies. And so understanding this phenomenon, it seems like it's very important. Maybe there are specific things older mothers should do that would be beneficial to offspring too.
We're starting to work with some reproductive biologists and developmental biologists. I'm not a developmental biologist, but that's obviously where all the action is happening. What it is these mothers are contributing or not contributing to their developing offspring as those moms get older. So I think that will help to provide some insights into sort of the reproductive aspects.
One of the interesting things we're puzzling over is we have some unpublished data here with collaborators around Singapore looking at methylation age in babies. It was kind of a secondary finding from the main project, which was to look at the adults in the population. But the interesting thing is it looks like the baby boys are already older than the baby girls. And so that I haven't wrapped my head around this yet, but it suggests something we already suspected and would be consistent with your data too, which is that aging is already happening, during very early in development. But it also leads to the question of what's different with the boys than the girls, and how does that relate to the maternal period?
Right. And when you're looking at those data, are you looking at maternal age?
But that's exactly what I was thinking when you gave your talk. We should go back and look at maternal age in that question too.
Yeah, that could really help take some of the noise out of the data.
Yeah. We and I wonder if the signal would be, there would be sex differences in the offspring in terms of the age they're born to. Yeah, it's a fascinating question. So at least I got an idea to go back and look at something. So
Okay, good.
that's but I think this is a great system, and there's a real movement these days toward trying to do everything very, very translationally and validate interventions and do things for humans. And of course, I'm very supportive of that, but this is just another one of these discussions that suggests that we also need to accelerate research on the basic discovery phases of aging research as well. I mean, I know you're doing drug screening, but there's some fascinating basic science questions that need to be asked in species like this. And the more that we expand sort of the longevity zoo, the more we're going to learn about mechanisms to modify the aging process. So let me bring in Anis Begum, who's a research assistant at the Center for Healthy Longevity. We have several questions that she can ask from the audience. Anis?
Hi, Dr. Kristin. Thanks for your presentation. That was really great. So we have one of the questions here that someone's asking. Have you checked how the lifespan looks if they cannot have offspring, comparing with the wild type? And are you comparing the offspring in the various mother age from the same mother?
Right. So the first part of that question, what happens if we prevent reproduction? Does that, say, increase lifespan, as has been seen in some other species? We've not been able to figure out a good way to prevent reproduction entirely. So we can decrease reproduction by, or almost eliminate reproduction by starving the rotifers, but that obviously changes lots of other things. We can use the chemical FUDR to prevent eggs from hatching, but really the reproduction has already happened. The rotifers are laying the eggs, so that's not really blocking reproduction. And we've not gone in yet with our CRISPR tools are quite new. so we haven't gone in with either RNAi or CRISPR to try to prevent reproduction using genetic manipulation. So it's a really good question, but we've not been able to address that directly. It is likely that there's, trade-off between lifespan and reproduction, as it's pretty classic evolutionary biology been seen across different species.
How much cell division is going on in the adult?
None, except in the germline. So they're like C. elegans that way. They're post-mitotic when they hatch.
So something like FUDR might have an effect that's used in C. elegans all the time.
So we do use FUDR. FUDR itself extends lifespan, but the rotifers are still producing eggs. I think those eggs are just not divided. There's no cell division in the eggs, so it's not really blocking reproduction.
The same thing, yeah.
Yeah.
Go ahead, Anise. Sorry, I'll let you go.
Yeah. And then the second part of that question was, sorry.
Are you comparing the offspring in various mother age from the same mother?
Yeah, it's the same cohort of mothers. So we start usually in our experiments, usually with 72 individual mothers, and we follow them over their whole lifespan, and we collect one offspring per mother at each of those, young, middle, or old ages. So yeah, same group of moms. So we're comparing siblings really across those different maternal ages.
Right, thank you. So the other question is, do you see any improvement in the mitochondrial function with the calorie restriction?
Yeah, we haven't looked at that directly, so I can't answer that question. I would guess that we'll see in rotifers what's been seen in other systems where this has been studied.
So there's also a similar question. How will the limited diet affect the mitochondrial activity?
Yeah.
No.
Yeah, it's a good question. Because so many other people are investigating caloric restriction, that's not something that we haven't gone into rotifers to try to look at mechanisms of caloric restriction directly. There's so many other laboratories doing really great work in that area already.
All right. So how does the genome look like in this animal, the rotifers? Does it only have one nucleus? Do you have any idea how they make sure the offspring got the right genetic information from the mothers?
Yeah, so these rotifers have a pretty standard diploid genome, 2 copies of each chromosome. The genome is relatively small. I think it's about 120 megabase pairs. So not huge, not tiny either. These rotifers, they don't have, a lot of intragenic, very large intragenic regions like you see in humans. And we think that in asexual reproduction, like I was saying, the development of the oocytes happens just by mitosis. So it's a pretty standard cell division process that leads to the full complement of the genome and a single copy of the genome going into each offspring. We haven't looked directly at mutation, either nuclear mutation or mitochondrial genome mutation, and how that might be increasing, how mutation rates could be higher in offspring from old mothers. There are multiple reasons to think that mutation is not really the issue. Part of that is that when we collect offspring from old mothers, and then from those offspring, collect their offspring when those mothers are young, we see a reversal of the negative effects of advanced maternal age when we switch back to young maternal ages. And so if there were really mutation happening in the nuclear genome, I'm not sure how we would see that switch back necessarily, correcting a mutation like that. So there's a whole other branch of rotifers, the deltoid rotifers. The ones I'm working with are monogonate rotifers. Deltoid rotifers have crazy genomes. They're degenerate tetraploids, so they're genome copied itself at some point, and then portions of that second genome kind of degrade over time. Those deltoid rotifers have also incorporated a lot of external DNA into their genomes over time. So they've taken in bacterial and fungal genes, incorporated them into their genomes, and then they're using those foreign genes, especially in stress responses. The rotifers I work with don't seem to be doing that.
Right.
How do they survive like extreme stress though? They're in these puddles, the puddles dry up, what happens?
Yeah, so the product of sexual reproduction is a resting egg, and that egg can be desiccated, it can be frozen. That's actually how we store rotifers, as we dry out their resting eggs and we throw them in the freezer, and they can hang out there for decades and decades. And then you pull them out of the freezer, put them in water, and rotifers hatch out in 24 hours. So they do have that resistant stage in their life cycle, like, African turquoise killifish or lots of other animals that live in these ephemeral environments.
Sorry, Anise, I just keep interrupting.
That's fine. So we have one more question. Are there senescent cells and cells?
I mean, it's a little hard to know in rotifers because they don't have cell division after they hatch. So in a way, all of their cells are senescent. They're no longer dividing, except in the germline. So that's not something we've really looked at in rotifers. Yeah, like rotifers are a good model for some questions, but definitely not for every question. Like similarly, we wouldn't study cancer in rotifers because they don't have cell division.
Right. So someone else also asked, can we also look at the difference in species of the organism used? Just wondering, how can genetic makeup of race with environmental factors affect the findings when relating it to the human conditions?
Yeah, that's a good question. That's something we've been doing in the lab. So we've published a couple of studies, one on caloric restriction and another on lifespan extension under low temperature, where we imposed those therapies across a dozen different strains of the same species. So basically different genotypes of the same species. And we see huge variation in across those different genotypes, both in response to caloric restriction and in response to low temperature. So I think that's a really powerful tool to, again, try to understand some of the basic biology. If we can take one strain that has increased lifespan in response to caloric restriction and another strain that actually has decreased lifespan under caloric restriction, and compare the gene expression or metabolism of those two strains, that can help us to understand, which mechanisms might be causative versus just, kind of along for the ride correlated with lifespan extension.
Yeah, that's the same observation that's been made in mice. I think everybody was surprised when it was made in mice, and then we've seen that in yeast too. We're using different genotypes. Except in that case, we were doing specific gene knockouts. I think that the challenge that everyone brings up is, how do you know you're doing the right level of calorie restriction? I mean, you can always argue that you're pushing some of the rotifer lines too hard, and then they're getting malnourished. So is it clearly true that you looked across a whole range and there's no way you can get extension?
Yeah, we looked across a small range of levels of caloric restriction. And we did see that, in our dozen strains, about a third had increased lifespan, a third no change in lifespan, and a third with decreased lifespan. So it's possible that, there's some other magic level in there that would increase lifespan even in those non-responding strains. But we didn't see that. It's a good criticism and potential issue.
I think probably I'm not doubting the finding. I think that's always been the question of these kinds of studies. Maybe you should try feeding them cheeseburgers. That'll probably shorten the lifespan. So.
Great.
Are there any other questions, Denise?
No, that was the last question. Thank
Okay,
you.
well, we're finishing a little bit early because we had the video that failed. So I just want to, first of all, thank you, Kristin. It's great having you on the show. Talk about something completely different, but very exciting, and probably going to provide major insights into understanding aging. So we'll look forward to seeing those. And thanks for coming on the show at an early time.
Yeah, thanks so much for.
Also, I want to let everybody know that we've decided to replay Matthias Roberts' presentation next week for the show. So you're going to have to wait 7 days to find out the end of that research project, which is pretty exciting. So come back next week for that. I want to remind people to use the chat function and the panelist and all attendees option to leave comments and feedback so that everyone can view the show. Our next webinar will be next week. I think I'll be back with Professor Jaap van Dien, Professor of Biomechanics at Vrije University in Amsterdam. I know I'm mispronouncing that in university. I'll get it right by next week. He'll be talking about feedback control of walking and how is it affected by aging and how we can maintain it. I want to leave you for a video about grandparents are 100% having more fun than you. Another year older, another year of fun. Thanks for joining the show.
Any closer though, I can't get the hula hoop in the picture, Sophia.
Like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.